Okay, hi everyone. So in this video we will be learning about the crosses that deviate from the Mendelian's inheritance. Okay, previously we have learned about the Mendelian's inheritance, where we have divided them into monohybrid and dihybrid. And for this particular part of chapter number 4, the genetic inheritance, okay, you should be able to explain briefly the types of inheritance that deviated from the Mendelian inheritance. There are 6 of them: the codominant allele, incomplete dominant alleles, multiple alleles, linked genes, sex-linked genes, and also polygenes. Okay, for the types of the inheritance, okay, we can divide them into two. Okay, whether the inheritance have single characteristic or they will have two characteristic. Okay, so the inheritance which have single characteristic, they will follow the Mendelian monohybrid inheritance. But there are some types of the inheritance which are deviated from the Mendelian monohybrid inheritance. Namely the codominance, the incomplete dominance, the multiple alleles, polygenic inheritance, and also the sex-linked genes. Okay, and meanwhile for the inheritance, okay, which have two characteristics, they will follow the Mendelian dihybrid inheritance. Okay, but the one that are deviated from the Mendelian dihybrid inheritance would be the linked genes. Okay, let's see the first one. For the codominance here, okay, it is the situation in which the phenotypes of both alleles are being exhibited in the heterozygote. Okay, if you still remember, we have learned about the homozygous and heterozygous. Okay, so the condition in which these two alleles of a locus are expressed in the heterozygote. Alright, so for example, we can discuss about the existence of the three different human blood groups called the MN blood groups. So there are three different human blood groups here. We have the blood group M, blood group N, and also blood group MN as the results of the codominance. Alright, okay, so if the individual they have the genotype of LM, okay, so this genotype they only have one type of allele, which is the LM only. So it will result in the phenotype of blood group M. Okay, for the second one, if the individual they have the genotype of LN, okay, so in this genotype they only have one type of allele, which is LN. So this will result in blood group N. But if the individual they have the genotype of LMLN, okay, so there are two types of allele you can find in this particular genotype, which is LM and also LN. Okay, so this will result in blood group MN. So meaning that both of the LM and also LN are being expressed in their phenotype. Okay, in other words, in the heterozygous, okay, they will be expressing both of the LM and LN in their phenotype. Okay, for the second part or the second type of inheritance which is deviated from the Mendelian is the incomplete dominance. Okay, so for the incomplete dominance here, it is a condition in which neither member of a pair of contrasting alleles is completely expressed when the other is present. Okay, in other words, heterozygous they have an intermediate phenotype between that of either homozygous. So the example that we can discuss for this incomplete dominance is the color of the snapdragon flower, or the scientific name is the Antirrhinum species. Okay, so for example here you can see the parental generation here. Okay, we have two different colors of the flower. It's either red or the white. Okay, we have two parents here, the red flower and also the white color. Okay, when we cross these two particular parent together, okay, the red flower we cross them with the white flower, right, they result in the first generation, the F1 generation. The F1 here means the filial number 1 or the first generation. They will result in the pink color offspring. Okay, different from the one of their parents. Okay, because the parents are having the red and also the white flower. But when we cross this red and also white parents together, okay, they will result in the pink color offspring. Okay, in their F1 generation. Okay, because of the mixture between the red and white color, so they will get the pink color. So that is what it meant by the intermediate phenotype, okay, between that of either of the homozygous. All right. Okay, so the heterozygous for color alleles, they will have pink color in contrast to the red and also the white color. Okay, for the red and also the white color. Okay, so the offspring will have the pink color instead. All right, so the red color is the dominant and also the white color is the recessive homozygous. Okay. Okay, for the F2 phenotypic ratio, for the monohybrid cross will then become 1 to 1 instead of the 3:1 ratio. Alright, so when we have this F1, okay, they are being self-cross. Okay, they are being self-cross together, meaning that the pink color of F1, we cross them with another pink color of the F1 generation. For the F2 generation, okay, they will result in the red flower. One of them is red flower, two of them would be the pink flower, and one of them would be the white color. So that is what it meant by the 1 to 1 ratio. Alright, phenotypic ratio. Alright, so this is the phenotypic ratio for the incomplete dominants which are deviated from the Mendelian. Alright, for the Mendelian, they will have the 3 to 1 phenotypic ratio. But for the incomplete dominants here, they will have the 1 to 1 ratio. Alright. Okay, the third deviation from the Mendelian inheritance is the multiple alleles. Alright, so for the multiple alleles here, it refers to one gene having more than 2 alleles and all of the alleles control the same characteristic. Okay, or in other words, 3 or more alleles for a given locus exist within the population. So this is according to Solomon. Alright. Okay, in chromosome number 9, okay, chromosome number 9, one part or one locus is controlling the blood type. Okay, we are for this case we are discussing about the ABO blood type group. All right. Okay, so if they have this IA, all right, it will result in the blood type A. If they have the IB here, they will result in blood type B, and if they have the IO, okay, the individual will have the blood type O. Alright, so I here is referring to immunoglobulin. Alright. Okay, so example for this multiple allele is the ABO blood type alleles which can be found in humans. Alright, so let's see the details. Okay, so there are 4 blood types for the ABO system here. Okay, we have the blood type A, blood type B, blood type AB, and also blood type O. Okay, so the ABO locus, they have 3 common alleles. Either they will have IA, they will have IB, or they also have the IO. Okay, IO can also be written as small I. Alright, so the small I is also representing the IO here. Alright, so the combination in ABO blood group system, okay, you will be finding these 6 different genotype, okay, for the ABO blood group system: IAIA, IAIO, IBIB, IBIO, IOIO, or IAIB. Alright, so for the IAIA and also IAIO, they will result in the similar blood type, which is blood type A. Okay. Meanwhile, for the blood type B, the possible genotype for the individuals with blood type B would be IB, or they can also have the genotype of IBIO. Okay, but different for the blood type O here. The only genotype that you can find for the individuals with blood type O is IOIO, or it can also be written as small I. Alright, and for the individual with the blood type AB, okay, the possible genotype for them is IAIB. Alright. Okay, so the 3 alleles for the ABO blood groups and their carbohydrates. So you can see here, okay, the alleles, we have the IA, we have the IB, also we have the small I here, also representing the IO. Okay, so IA, they have their own specific carbohydrate. Okay, for the A, similar with B, they have their own specific and unique carbohydrate for B, but for the IO or the small I, there are no carbohydrate can be found on that particular blood group, red blood cell. All right. Okay, if we see on their genotypes and also phenotype here. Okay, for the genotype IA or IA small I, okay, also can be written as IAIO. Okay, the red blood cell appearance would be looking like this. Okay, so you can see, for example, if we represent the carbohydrate for A with a triangular, all right, so it will be surrounded, the carbohydrate, the triangular carbohydrate can be found on the red blood cell of the phenotype blood group A. Okay, for the blood group B, okay, if we represent the carbohydrate for B as a circle carbohydrate, okay, those with the genotype of IB or the IB small I, okay, you can see the red blood cell appearance here, you will be finding the circle carbohydrate surrounding them. Alright, so this is the appearance of the red blood cell for those individuals with the phenotype blood group B. Okay, and for the individuals with the phenotype of blood group AB, okay, the genotype, the only possible genotype would be the IAIB, where you can see the appearance of the red blood cell will be a mixture between the carbohydrate of A and also B. Okay, you can see the mixture of the triangular and also the circle here on their red blood cell. All right, so that is how the AB blood group red blood cell will appear. All right, but different case for the individuals with the genotype of small I also can be written as IOIO, right? Where they result in the phenotype of blood group O. None of the carbohydrate A or B will be found on the red blood cell. Okay, meaning that there will be no carbohydrate can be found on the red blood cell of the individuals with the O blood group. All right, so that is the case for the multiple alleles. Okay, for the next one, for the polygenes, okay, the other types of inheritance which are deviated from the Mendelian, okay, the polygenes, also known as the polygenic inheritance, okay. So for the polygenes here, it is an additive effect of two or more genes on a single phenotypic character, okay. In other words, multiple independent pairs of genes have similar and additive effects on the same characteristic. Okay, so many genes are controlling the similar character. Okay, for example, the characteristic that we can use to discuss about the polygenes here are either the height or the skin color. Okay, so for the polygenes here, all right, it occurs when a trait is governed by two or more sets of allele, possibly located on many different pairs of chromosomes. So this is according to Meda, okay, in his 12th edition of book. All right, so you can see here we have three different letters representing the three different alleles, A, B, and C. But all of these A, B, and C are controlling one particular character. For example, just now, okay, you can use the height, or you can also use the skin color, okay, to discuss about the polygenes here. All right, so this polygenic inheritance could result in a bell-shaped curve, also known as the normal distribution curve. All right, so you can see here, okay, this is an example of the skin color. All right, which give off the results of a normal distribution curve. All right, a bell-shaped curve. Okay, in details for the polygenes here, if an big A, big B, big C person would be very dark, okay, in opposite, a person with small a, small b, small c, small c genotype would be very fair. All right, so this is what we call as the homozygous dominant person. All right, meaning that the dark is the dominant characteristic, and the fair would be the recessive characteristic. All right, so this is representing the homozygous dominant, this is representing the homozygous recessive. All right, but for those who are heterozygous, okay, the heterozygous person, for example, we have here big A small a, big B small b, big C small c. All right, this is a heterozygous genotype. That person would have a skin of an intermediate shade. All right, so it is different from this person with the homozygous dominant and also homozygous recessive color, skin color. All right, okay, because the alleles have a cumulative effect, the genotypes big A, big B, big C, and also big A, big B, and small c would make the same genetic contribution, okay, to the skin darkness. All right, so you can see these two different genotypes are actually heterozygous, okay, and the 3 units here will be controlling the intermediate skin darkness. Okay, for the next genetic inheritance, which is deviated from the Mendelian genetics, is what we call as the sex-linked genes. Okay, for the sex-linked genes here, it is the gene which is located on either of the sex chromosome. All right, so this is according to the Campbell, and most of the sex-linked genes, they are on the X chromosome and show the distinctive pattern of the inheritance. All right, so it is the gene which carried on the sex chromosome, and in mammals, almost all of sex-linked genes are located on the X chromosome, or in other words, we can call them as the X-linked. All right, so there are two genetic conditions that we can give the example for the sex-linked genes here, which are the hemophilia and also the colorblind. Okay, when we talk about these sex-linked genes, right, we will be comparing between the males and also the females. All right, for the human females, they have two X chromosome, two X's here, which means that they have two sex-linked alleles. Right, and for the human males, they have only one X chromosome, and the other one would be the Y chromosome, okay, the XY, which means that they have only one sex-linked allele. All right, so in other words, that these males, they are heterogametic. Okay, they have the XY chromosome on their sex chromosome. All right, and for the females, this is what we call as the homogametic, because they have both of the X chromosome being the X's, right? So X and X here. All right, so this is why males suffer from the effects of the sex-linked genetic diseases more often compared to the females, because they only have one X chromosome which can be easily affected. Okay, and there are no non-Y linked traits, probably because the Y chromosome, they carries so few genes. Okay, now moving on to the linked genes, or the gene linkage. Okay, for the linked genes here, it is referring to two or more genes which are located on the same chromosome. All right, so these genes, they are located close enough together on a chromosome that they tend to be inherited together. All right, so when they are inherited together, they will not obeying the Mendelian's law, right? Because they will not undergo the independent assortment. All right, so the tendency for a group of genes located on the same chromosome to be inherited together in the successive generation. So this is according to Solomon, okay, for the definition of the linked genes. Okay, for the linked genes here, okay, since they are the genes that are on the same chromosome, okay, they are linked together. All right, so such genes, they do not obey the Mendelian law because they do not undergo the independent assortment. Thus, they are inherited together unless they are separated by crossing over during prophase 1 of the meiosis. Right, so if we see in this table, okay, we are comparing between the dihybrid and also the linked genes here. Okay, so the genotype for this one is the heterozygote. All right, and after the process of meiosis, okay, the gametes that the dihybrid they are producing, there are 4 types of gametes here. All right, so we have the big A and big B, big A and small a and big B, and small a and small b. All right, so there are 4 types of the gametes produced, okay, from the meiosis due to the results of the independent assortment. All right, Different cases for the linked genes here, okay, because they are linked together. All right. So when they are linked together, okay, meaning that they will not obey the Mendelian's law. All right. They will not undergo the process of independent assortment. All right. So if they are not undergoing the process of independent assortment, okay, you can see from here the gametes that they are producing, instead of 4 different types of gamete, they only produce 2 types of gamete here. All right. So you can see here these two offspring, okay, they have similar gametes here, right? They have the big A and big B, big A and big B, right? So that is one type. So another type of the gametes they are produced from the linked genes here is the small a and small b. All right. So this is the results of the gametes which do not undergo the process of the independent assortment. Right. So that is the case for the linked genes. Okay. So for the linked genes with crossing over, okay, if you refer to this table, okay, when we do the test cross for the F1, okay, the first generation, they are being crossed with the homozygous recessive, okay, the phenotypic ratio will not be 1 1 1 1, okay, in comparison to the Mendelian dihybrid inheritance. Okay, for the linked genes here, okay, the phenotypic ratio will not be 1 1 1 1. Okay, but for the genotypic ratio, it will stay as 1 1 1 1 ratio. Okay, and for the last part of this genetic inheritance, chapter number 4 would be the genetic mapping. All right. Okay, for this genetic mapping, the first thing that you need to do, that you should be able to do, is to define what does it mean by the genetic mapping. Okay, and the second one is to identify the position or the order of the genes along the chromosome based on the recombinant data. Okay, for the genetic mapping, it is the ordered list of genetic loci along a chromosome. All right, so it is determined by the percentage of recombination, okay, or the recombination frequencies, which we call as the linkage map. All right, so this is according to Campbell, right? And the percentage of recombination, they can be obtained by calculating what we call as the crossover value, or simplified as COV. For the COV, or the crossover value formula, okay, what you can do is you take the total number of the recombinant. Okay, you take the total number of the recombinant and you divide them with the total number of the offspring. Okay, and then don't forget to times them, or you need to multiply them with the 100. All right. So for the recombinant here, it is referring to the offspring which have different phenotype from their parent. Okay, meaning that they do not have the phenotype which is original from their parent. So that is what we call as the recombinant offspring. Right. So for the COV here, what you need to do, you divide the total number of recombinant, divide them with the total number of offspring, then you times them with 100. Okay. If the percentage of the recombination, okay, the value of the COV value here is large, okay, around 20 to 40%, the genes are relatively far apart on the chromosome. Okay, since they are far apart from one another, there is high possibility for the crossing over to occur. Okay, the process of the crossing over, they have high possibility to occur. Alright, but if the percentage of the recombination, the COV value, is low, around 1 to 5%, okay, the genes, they are relatively close together on the chromosome according to their COV value. All right, so since they are very close together, there is lesser possibility for the crossing over to occur. Okay, and the distance between the genes on the particular chromosome is expressed in map units. Okay, so when you give the value of the COV, right, so you can label them as the map units. Okay, and one map unit is equivalent to 1% of the percentage of recombination. Okay, and also today the word centimorgan is often used. All right, so there are 3 different units that you can refer for the genetic mapping here. You can label them as the map unit, you can also give them in terms of the percentage, or you can give in terms of the centimorgan unit. Okay, and in conclusion, through this genetic mapping, we can determine 2 things. Okay, the first one is relative distance between these two genes, and the second one is the linear sequence of the linked genes on the chromosome. All right, that's it from me. Thank you for listening.